Welcome to Faculty Voices. Noreen Tuross is Landon T. Clay Professor of Scientific Archaeology at Harvard's Department of Evolutionary Biology. Her most recent project is A Land of Art and Botanicals, collaborative research at the Cueva de las Manitas in Oaxaca, Mexico. Welcome, Professor.
Thank you. Thank you for being interested.
Could you give us a really quick overview of your project, especially for those of us who aren't involved in the scientists?
This project was an opportunity to once again work with a wonderful archaeologist in Mexico, Dr. Nelly Robles Garcia, who is centered in Oaxaca and has done many projects over the years. Nelly was excavating a new cave, a cave that had not previously been excavated in Oaxaca. And what's important to realize is that much of our knowledge and impressions about early Mexico come from caves that run down the center of Mexico in both Puebla and Oaxaca. They've been the source of archaeological investigations for, let's see, 60, 70 years, primarily led by Americans. Niche is very famous for excavating many of these caves, but more recently, Mexican colleagues have started re-excavating and reanalyzing this material. It's important because it forms a lot of our baseline information and knowledge about domestication of plants in the Americas, which is essential for understanding past lifeways in the Americas. People of Mexico and other countries, including many in South America, were experts at domesticating plants and using them as their food base. So the interest in these caves comes from the fact, in part, that they are extraordinarily well preserved. They're called dry caves, and the material is exceedingly well preserved.
The title of your project is A Land of Art and Botanicals. You've just talked to us about the botanicals, but you're a scientist. Why art?
What sets La Cueva de las Manitas apart from many, if not all of the other caves, is that it's an artist's showroom. The entire surface of the roof surface is covered with rock art. And that was really what led Dr. Robles Garcia to the site and what piqued my interest. The reason that you put up with having a scientist involved is it's incredibly difficult to date, to form an accurate chronology of rock art. When did the people paint the images? And from first looks at the cave by people who are far better at looking at images than I am, it was pretty clear that this was not a single event, that this was an artist's cave. This is where people came back time and time again to paint. And what was really fortunate for us is that the artists left some of their tools in the sediment. So there are paintbrushes made of agave that were excavated in the site. There are other things like stylus, but there are multiple brushes. So unlike any place I've ever heard of, you could date the art from the hand of the painter. He or she left their tools that were vegetable matter that are amenable to C14 dating in the cave. And so that formed the basis of our investigation.
What did this art look like?
So it's called Manitas, which they have translated as little hands. So there's classic handprints that you see, really fascinating, all over the world. There are also animal figures, more traditional ritual figures. There's a wide variety of art that has not yet been completely described, but will be. That's another aspect of the work that's ongoing to describe this artwork.
Do you remember what you felt when you first saw this rock art and when you discovered this agave brush?
I wasn't there when the excavation was done, but when I was shown the material, sometimes archaeologists show you things and they say, see, it looks like this, and you go, really? This was unambiguous. This looked like a paintbrush. If you had a piece of agave and you wanted to make a brush, it would look just like this. So it was astounding actually to see this material. In terms of, say, I've seen pictures of the images. What's amazing is how wide and complete they are. This isn't a single image. They use the entire canvas of the cave, which is really a rock shelter, to paint. So unlike a lot of the other caves that seem to be living surfaces where people went to cook or to collect or to live, this really does seem to be an artist's cave.
Huh, so it's an art gallery.
It's an art gallery, absolutely. And so that was the title. That brought the art and the botanicals together because it was the material that they left behind and used to make the paintings were botanicals. And as I've indicated, the history of domestication and use of botanicals in Mexico is second to none. They were excellent gardeners and botanists and users of plants.
Besides the agave brush, were there other intersections between these artistic endeavors and plant utilization?
So there was at least one stylus. It looked like a stylus, a sharp pointed object that you could probably make dots on, but the ones I remember were the agave brushes. Now, there was lots of other plant material in association with the brushes that we also radiocarbon dated. And so that really enlarged the interpretation of the cave and let us ask the question, not only what was the date of the brush, but what was the most intensively used time in the cave? In other words, when you radiocarbon date, we've done now 48 radiocarbon dates, which for the size of the cave is an enormous number of radiocarbon dates. When you look at the range of radiocarbon dates, you can see timelines. You can see intensity in the timeline where the cave was used. And about 2200, 2300 years BP, there was an intense use of the cave, but there were other eras where it was also used.
You mentioned the process of radiocarbonate. Could you explain that for myself and our non-art scientific listeners?
When anything alive is growing, it is taking up carbon, as we know. And when the plant or the animal dies, the uptake of carbon stops, and the amount of radiocarbon in that plant or animal starts to decay because it's radioactive. And so by looking at the amount of radiocarbon in a sample and comparing it to calibration curves that have been established, you can come up with a really accurate date of the material. And if you date enough material, you can actually really hone in on an exact date and also, as I indicated, the intensity of the usage of a site. So you can date plant material, and in this case, we were dating both plant material and charcoal. And charcoal is an excellent source for radiocarbon. Animal material, it's generally the bone collagen that is dated. We don't have any animal material in this cave.
Mm-hmm. How did your research this summer advance your project, advance your findings?
We finished all the radiocarbon dates. We finished what we had said we would do. And now we're in the process of analyzing it all. And one thing I want to do that will take some time is put it in the context of these other caves that have been studied and excavated over the last 60 years. Where does the timeline fit in? Is it different? And so that's going to take a fair bit of analysis, but the proposal indicated that we would date this material, and that has been done.
Did you meet any unexpected obstacles?
No.
That's great.
This is one of the simplest projects I've ever done, again, because the material is so remarkably well preserved. You have to do is get permits. We jump the hoop of getting proper permits, both to take archaeological material out of Mexico and to bring plant material into the United States. So if there were hoops to jump, but those are just hoops, you just do it. I said we have 40, I believe we have 48 radiocarbon dates. We took 50 samples. That means only 2 samples didn't produce enough datable material. That's extraordinary. We haven't encountered any real difficulties. I have to say that one of the satisfying things about this cave is that it's small. It's important, it's an artist place, but it's small. So you're not faced with meters and thousands of samples to choose from. So we could do a fairly broad reach analysis of the cave because it's small and very well intact.
When you say it's small, is it like somebody's kitchen? Could you lie down in it 3 times? Could you give us some idea of the dimensions?
I just read the dimensions. So I think it's fairly wide across in terms of tens of feet, but very shallow, maybe. I don't actually know, but it's like a slice through the landscape. But when I say small, it's actually less important what the dimensions of the rock shelter are and how deep the sediments go. And here the sediments only went 1 meter deep and only 1 meter in a very small part of the cave. So there wasn't much deposit by ancient humans in the cave. They just didn't bring a lot of material in there. And then that's opposed to other caves in the area where you get 3, 6, 7 meters worth of material to excavate.
But was that an advantage in the sense that it was easier to find things, or was it a disadvantage?
For me, it was an advantage. I felt we could really take a holistic view. Sometimes archaeologists like a deeper sediment, they will find more stuff, but in terms of excavating, this was excavated at the centimeter level, centimeter by centimeter. So you can say it was only a meter there, but if you're going centimeter by centimeter, it's still a job. That's the archaeologists on Dr. Robles Garcia's team are just excellent. They're excellent archaeologists, and it's a pleasure to work with them.
What does the local collaboration bring to your research?
Everything. My collaborators know so much more than I do, especially about the region, about other types of projects that are going on. They know the literature. It's fascinating. I go down for a week and I learn a year's worth of knowledge. So it's wonderful.
How did you get involved in doing this investigation in Mexico?
So I've worked with Dr. Robles Garcia previously, and it's always been a joy. It hasn't always been this straightforward and this easy, but I think if you work across disciplines and you find people who are simpatico, who you get along with, who you admire and you trust, you should stick with it. And I've stuck with a couple of, including Dr. Robles Garcia, and have enjoyed it tremendously.
How did you first meet her? What was that initial contact like?
Oh, that's interesting. I actually met Dr. Robles Garcia through my husband, he was an archaeologist for the National Park Service, and he had met her on actually heritage sort of issues. In addition to being a real dirt digging archaeologist, Dr. Robles Garcia is an administrator and also has a real expertise in heritage preservation, saving sites and monuments. And so my husband had met her. We went down to Mexico, and I think it's been maybe 15 years that we've
Wow,
been working together.
15 years of collaboration. What other projects have you worked on with her?
We worked on something called Tomb 7 at Monte Alban. We did a project in Tepozcalula looking at a contact site. So there've been a variety, but this one is completely different. And I have to say, I've really enjoyed this.
That's an amazing thing to have this different kind of experience after you've been working in the field for so long.
It is. It is. I wish I tried it earlier, but they don't come along all that often. But people find a new cave and do a thorough excavation and involve, and I'm certainly not the only other person involved. There are experts looking at the pigments, there are experts looking at the images. So it'll be a nice story to add to Mexican history when we finish.
That is absolutely fascinating. I'm also fascinated by the fact that of gender, that you're working with a female archaeologist, you yourself are a female archaeologist. How does gender play into this?
You keep your head down and keep working. And I have enjoyed working with a female archaeologist. I'm actually not an archaeologist, I'm a biochemist. Again, it's the more dramatic thing is working across fields. That's actually much harder than working across gender. But you're right, female archaeologists in Mexico, probably anywhere, are few and far between. And I hope there are more in the future.
You're a biochemist, but you're
Yeah.
also the Landon Clay Professor of Scientific Archaeology. Do you want to explain to me?
I'm not sure why they hired me, but I'm actually the Landon T. Clay Research Professor. I have just recently retired. But this is not uncommon now in archaeological, ecological endeavors that you need people across disciplines and you need people with different technical expertise. And that's how I've ended up in the position.
That's fascinating. When you became a biochemist, I imagine that was a fairly unusual field for a woman.
So actually, when I trained at my PhD and my postdoc, it was not unusual for women. I was always in co-ed situations. It was always pretty straightforward and pretty easy. I know, not the usual tale, but that was my experience, both in my PhD and I was a postdoc at the National Institutes of Health, which was like science heaven, and a group of wonderful people who trained me and put up with me.
Uh-huh. But how did you get into the archaeology part? When did that
Oh,
happen?
because I'm a bone biochemist. So right from the start, I was interested in if I wanted to describe one thing that interests me, it's in how things fall apart. And because I had training in bone, people who are interested in bones falling apart they're archaeologists, but they're also paleontologists and ecologists. And more broadly now, I'm interested in how things fall apart and why they fall apart and why they don't fall apart. Because everything falls apart. Everything organic falls apart. If you want something that doesn't fall apart, you should study lithics. You should study stone tools. They hold up pretty well.
That's fascinating. I love this piece of, when was the first time that you actually worked on an archaeological project?
The first time was during my, oh no, actually I did some work on my undergraduate, but then a project in my postdoc, and it wasn't archaeological, it was called taphonomic. It was how animals who had died naturally and how their skeletons were decaying and the change that happened through time. That project was led by Anna K. Berensmeyer, who's still at the Smithsonian, and another wonderful researcher. It was my honor to work with her.
That's absolutely a wonderful history. Going back to Mexico, do you foresee, I know you say that you just took retirement, but do you foresee yourself working on any other caves or any other projects in Mexico?
I always say no, and then Nellie comes up with something just wonderfully interesting. But I think any involvement in large project is at an end for me. I'll always be interested, and if I can help out, I will. But as one of my colleagues said, there's time to hang it up and go fishing. I think I'm at that stage.
Okay, I really enjoyed hearing about the Mexican cave and a little bit of your life history. This is Faculty Voices. You've been listening to Noreen Tuross. She's Landon T. Clay Research Professor of Scientific Archaeology at Harvard's Department of Evolutionary Biology. And her most recent project is A Land of Art and Botanicals collaborative research at the Cueva de las Manitas, Oaxaca, Mexico. It's been a pleasure talking to you.
Thank you so much.
Thank you.